This is one of the lectures for week 8, and it's covering chapter 37, the endocrine system. This will be very important moving into lab next week, because next week in lab you'll be talking about homeostasis. We'll be talking about different feedback loops in lecture today that contribute to homeostasis or move us away from homeostasis, and we'll talk a lot about some of the signaling and regulation that's involved in these bigger processes. I'm also going to cover a little bit of anatomy that we're not scheduled to cover in lecture for a few more weeks, but I think it's important for you to at least get a review of it before you go into lab next week. So keeping in mind from previous weeks, we're thinking about chemical structures, both in terms of whether a structure is hydrophilic or hydrophobic, and then also the structures of key macromolecules like carbohydrates, proteins, fats, and nucleic acids. I also want you to keep in mind cell signaling and receptors. So again, this idea that cells can communicate with one another through these chemical pathways is really the crux of what we're talking about today. Keep in mind the structure of the plasma membrane, which parts of it are hydrophilic, which parts are hydrophobic, and I'll remind you of that. And then also diffusion and osmosis. And again, this is all to prepare you for the homeostasis lab that you're going to be doing next week. So the sections that we're covering in this order are section 1, which is about the types of hormones. So you should really focus on the 3 kind of broader categories of hormones based on their chemical structure. There's a lot of different ways that we can classify hormones by function, but we're really going to be focusing on broad chemical structures today. Then we'll get into how hormones actually work, so thinking about where they bind to receptors. So you should know where we would expect to find those hormone receptors. And then I'm going to switch the order up a little bit. We're going to go straight to section 4 and talk about hormonal regulation, so different negative and positive feedback loops. And then also 3 different mechanisms that stimulate hormone production. We'll get into the actual endocrine glands, which is where hormones are secreted from or produced in. So you should know the names of the glands, but you don't need to know what specific hormones are produced in each of those glands. So that's more of an anatomy and physio requirement, at least in terms of lecture. And then for section 3, we'll come back to that at the end and touch base specifically on the excretory system. And when we mean the excretory system, we're talking about the formation of urine and the filtration of our blood. And so there's other stuff in that section about digestion and about reproduction. We'll cover those in different lectures. Okay, so the endocrine system really comes down to chemical signaling, and there's a lot of different types of chemical signals in our body, a lot of different classifications. Here we're specifically talking about hormones, which are chemical signaling molecules. They're secreted by the endocrine system, and they have an effect on a target cell somewhere else in the body. So we talked before about endocrine, paracrine, and autocrine signaling. I'll review those for you today. This hormonal response may be rapid or it might be slow. Oftentimes it's kind of a slower response. We'll talk about one exception to that today. Hormones have a lot of different effects, and the same hormone can have many different effects depending on where it is and the relative dose of it. But these are all carefully regulated by feedback loops. Next week, we're going to be talking about the nervous system. And so the nervous system involves both electrical and chemical signaling, and it's a very fast response to our external environment. We can talk about the endocrine system a little bit differently. The endocrine system is chemical signaling only. There's no electrical component to it. That being said, the nervous system does interact with the endocrine system and regulate it in some ways, but when we're just talking about the endocrine system, that's just chemical signaling, and it's more of a slower response to our internal environment, so kind of looking at the balance inside of our body and trying to maintain what's called homeostasis. So there's different structures within the endocrine system, and when we talk about those endocrine glands, what we mean are ductless glands. So when we say ductless, we mean that they don't have a tube or an opening that goes out into our external environment. So for example, we have salivary glands that produce saliva that travels through a duct and then it goes into our mouth, which is technically the external environment. We'll cover that more in digestion. We have tear ducts that form where the tears that are formed kind of in glands within our eyes are secreted out of. So that's called exocrine glands when there is a duct that takes something out of our body. The endocrine system has endocrine glands, so not exocrine, but endocrine, where they're secreting those hormones directly into the surrounding fluid. So they're not leaving our body, they're not traveling through our duct. So some examples of that include the pituitary gland, the thyroid and parathyroid, which are pictured to the right, the adrenal gland, several others that we'll talk about in a little bit. So like I mentioned, we've reviewed different types of chemical signaling. Autocrine is signaling within a single cell. So remember, auto is self, like an autobiography is a book about yourself. An autocrine signal is signaling sent from a cell to that same cell. Paracrine, para means nearby or close. So paracrine signaling is signaling between cells that are close to each other. Endocrine signaling is signaling across great distances within the body. So for example, the brain sending a signal to tissue in the kidneys, which we'll talk about actually later in this lecture. So when we're thinking about hormones and how to divide them, I mentioned it's kind of tricky to divide them based on function, but one quick and easy way to divide them is based on their chemical structure. And that's useful because it tells us a little bit about how they travel through our body and how they bind to and have an effect on cells. So we have lipid-derived hormones, we have amino acid-derived hormones, and we have peptide-derived hormones. Peptide and amino acid are going to share a lot of chemical properties because amino acids make up peptides, which we'll revisit in just a moment. So we'll walk our way through each of these three examples and talk a little bit about their chemical properties and what types of hormones you would expect to find in each of these categories. So for lipid-derived hormones, this is an example of the chemical structure of estrogen. You've probably heard of it. It's a very classical sex hormone, and we'll talk about it a lot more in the reproduction lecture. So these lipid-derived hormones tend to be cholesterol derivatives. They are similar in structure to cholesterol, which is an important component of the cell membrane. These steroid hormones include sex hormones like estrogen, which I mentioned. Estrogen's kind of a broader class, so estradiol is considered an estrogen, as well as testosterone, which is an androgen. Androgen means it's typically associated with the male reproductive system, and we mean that in the anatomical sense. When we talk about reproduction, I'll talk about kind of different biological distinctions of sex classification, including hormonal and chromosomal and anatomical, and how that all kind of is distinct from gender, which is a big conversation. And then also cortisol is a steroid hormone, and cortisol is associated with stress. So these lipid-derived hormones are cholesterol derivatives, and they are hydrophobic. So as fats, as kind of fatty components, they are insoluble in water, which means that they can't float around in your bloodstream by themselves. They can't float around in the interstitial fluid by themselves. They have to be picked up by proteins, these helpful carrier proteins that keep them safe, keep them moving around in blood, and allow them to bind to the cell membrane. And actually not just bind to the cell membrane, but move through it. So we have lipid-derived hormones, which are hydrophobic, and then we also have amino acid-derived hormones, which are hydrophilic. So these are individual amino acids. Amino acids have a central carbon, a hydrogen. They have an amino group, which is a nitrogen and 2 hydrogens, a carboxylic acid group, which is a hydrogen, 2 oxygens, and a carbon, and then they have an R group. And for every amino acid, there's a different R group. Tyrosine and tryptophan are 2 amino acids, so they have unique R groups. And when we modify these amino acids, these individual amino acids, we get amino acid-derived hormones. So these are things like melatonin, which is pictured here, as well as epinephrine and norepinephrine. So these are water soluble, which means they can easily travel around in our circulatory system, in our interstitial fluid, they can move around in our cytosol, but they might have some issues getting across the plasma membrane, which we'll talk about shortly. Closely related to amino acid-derived hormones are peptide-derived hormones. So remember, when we have individual amino acids, those are held together with peptide bonds. So when we have a short chain of amino acids, that's a polypeptide. And so a peptide is a shortened term for a small protein, so a short chain of linked amino acids. And then when we have a bigger chain of linked amino acids that might be folding in unique ways, that's a protein. So there are peptide-derived hormones that are peptides, which are small proteins, and proteins, which are full proteins. So these are often stored in vesicles within the cell and released as necessary. And just like the amino acid-derived hormones, these are water soluble, they're hydrophilic, which means that they're not going to be able to move through the plasma membrane. They're going to have to bind to receptors on the outside of the cell. You've probably heard of insulin before. You might have heard of oxytocin, which is considered the love hormone. It's associated with bonding and it's released during birth and breastfeeding and physical contact, different things like that. And there's also antidiuretic hormone, which is called ADH, and we'll talk about that quite a bit more during lecture today. So we've talked a lot in this class about signaling molecules, how a ligand, a signaling molecule, might bind to a receptor and then have an effect if the receptor for that ligand, for that signaling molecule, is found on a cell. So far we've kept it kind of simple. We've kind of been like, either the receptor is there or it's not, but it's not that straightforward. So a cell can have many receptors, it can have many of a particular type of receptor, and it can actually change the number of receptors that are present. And all of that affects the effect that it has on the cell, so the cell's activity. So remember, sometimes a receptor is present, sometimes it's not, but again, it's not that straightforward. We can change the number of receptors. Either way, the goal of having a receptor and having it bind with a ligand, which might be in this case a hormone, is having a response, having an effect within the cell. So I want to point out here in that central target cell, there's 3 arrows leading to the response. The implication there is that it's not just a one-off process. There's a lot of steps that have to take place in order to have that response. So you don't just magically have the hormone binding to the receptor and then the cell does exactly what you want it to do. That complex has to kind of move around in the cell, lead to a series of chemical reactions, maybe lead to some changes in protein expression in order for that to have the response that you want. So again, there's signal perception, intracellular signal transduction. There, when we're talking about signal transduction, we're talking about kind of propagating and sending along that signal through a series of steps before we get to the ultimate response. So this picture has a lot going on and a lot that we could unpack. I'm going to leave it here so that if you want to go back to it and study and compare kind of intracellular receptors versus extracellular receptors all in one slide with a lot of detail, you can. But in general, you should know that hormones bind to receptors, which might be located inside the cell, so intracellular, or fixed onto the cell membrane, which are extracellular. So I'll walk you through each of those in turn. With intracellular hormone receptors, this is very useful for hydrophobic hormones. So hydrophobic hormones, which are lipid-derived, can quite easily pass through the cell membrane and then bind to receptors. And the receptors are inside of the cell, so they're floating around the cytoplasm. They may even be inside the nucleus, so that receptor or the molecule is able to pass through and eventually get into the nucleus. So remember, when we have DNA inside of the nucleus, that acts as a code for RNA through a process called transcription, and then that messenger RNA acts as a code for proteins through a process called translation. So when we have transcription factors or transcription regulators, these increase or decrease the production of messenger RNA using that DNA code by binding to DNA. So if you think back to one of the earlier reflections we did, I had you think about transcriptional regulators or transcription factors. So different hormones, specifically intracellular hormones, are an example of transcription factors. They can work their way into the cell, hook up with DNA, and either encourage or prevent the reading of that DNA code in order to produce proteins. So there's also extracellular hormone receptors, and these are particularly useful for those hydrophilic protein kind of associated hormones. So these are also called plasma membrane hormone receptors because they are hooked into the plasma membrane. So in this image, you can see the hormone epinephrine binding to a receptor called beta-adrenergic receptor. So it's able to bind to that external extracellular hormone receptor. So again, when you're learning these words, don't get overwhelmed by the number of terms. Think about what it's telling you. Extracellular, extra means outside of, cellular is the cell. So these are hormone receptors that bind to the outside of the cell. And these are very useful for hydrophilic or water-loving hormones, because if you look at that plasma membrane, there's a ton of fat running through the middle. There's those lipid bilayers, and so those hydrophilic, water-loving hormones would not be able to pass through that. So they have to bind to extracellular hormone receptors that are on the outside of the cell. So when they do that, they activate different signaling pathways. Remember, it's not just a one-off reaction. So we call these extracellular hormones or these water-soluble, water-loving hormones first messengers, because they don't make their way directly to the DNA or directly to where the effect needs to be. They lead to a signal pathway. One example of a second messenger that kind of receives that signal from the hormone and continues it along is cyclic AMP or CAMP. So you might hear about that a lot in future biology education. It's a very important signaling molecule for sending signals within the cell. So we also talked about how we can kind of change the number of receptors that are found on the cell. That's called upregulation if you're increasing the number of receptors, and downregulation if you are decreasing the number of receptors. So kind of changing the intensity of the effect. And so the number of receptors that are present helps have an effect on how sensitive the cell actually is to that hormone, whether it's able to pick up kind of the intensity, and then also it has an effect on the cellular response, because if you have more signals being sent in, you're having more effects and more activity within the cell. So upregulation increases the number of receptors and increases the cell sensitivity to the hormone. That in turn increases the activity that is happening inside the cell as a result of that hormone. Downregulation decreases the number of receptors, and it also decreases the cell sensitivity to the hormone, which in turn decreases activity. Okay, so I've mentioned that hormones are very important for this idea of homeostasis. And if you think about something being in stasis or static, that means that it's kept steady or fixed. So it's kept still. And so homeostasis refers to this idea that there's kind of a steady state within our body. It's not exactly perfect. So think about your body temperature. It should be about 98.6, but if you were to use a thermometer and it was about 97.5 or even 99.1, it wouldn't be that big of a deal. You wouldn't go to the doctor. So even though there's kind of a steady state, you still have a range at which it's acceptable. If you go too far outside of that range, then you initiate these kind of bigger changes. You kind of trigger these feedback loops to keep things consistent. So it's kind of like a seesaw. If you go too far in either direction, you get pushed back through this series of effects. And the endocrine system is really important for kind of having an effect. So we might have the nervous system kind of sensing whether things are being kept in the correct state, and then we have the endocrine system enforcing that state. So there's a couple of different types of feedback loops. There's positive feedback loops and negative feedback loops. I really want to encourage you in biology never to use the words positive and negative as good or bad. Positive always means more of something, or it might mean something's present. Negative means less of something, or it means something is not present. They don't mean great or bad. And in fact, negative feedback loops are really good for us. They help us maintain homeostasis, and you should for sure know that. You need to know that negative feedback loops are the way that we're able to maintain homeostasis. Positive feedback loops keep us out of homeostasis, so they move us away from homeostasis. They get us to an end point. So for example, a positive feedback loop is labor and delivery. You have the fetus pressing against the cervix. That's sending a signal to the brain to secrete more oxytocin, which causes stronger contractions and pushes the baby more against the cervix. So you have the effect resulting in more stimulus. And so that goal is to get the baby out of your body, which is moving you out of homeostasis and getting you towards an end point. So that's a positive feedback loop. A negative feedback loop, the effect results in the end of the stimulus. So for example, if your body temperature drops, you might start shivering, you change your blood flow, different things like that. So you start retaining more heat and you get back to normal body temperature and you stop shivering, you stop changing your blood flow. If your body temperature rises, you change your blood flow in other ways, you start sweating, you go through evaporative cooling, you cool down, and then you go back to your normal body temperature. You wouldn't keep sweating. That would be really dangerous for you. So a negative feedback loop, the effect results in the end of the stimulus. And again, please make sure you're not thinking about these as good or bad. Negative keeps you in homeostasis, positive moves you out of homeostasis. So when we're thinking again about this idea of regulation, there's a few different things that send signals to our endocrine glands to synthesize and secrete hormones. So the endocrine glands make the hormones and they also release them when the time is right, when they receive a signal. So that signal might be humoral, it might be in the extracellular fluids. Humors is just kind of like an old-timey word for body fluids. And so when there's kind of changes in like our ion concentration in our blood, that's a humoral signal, a humoral stimulus. There's hormonal stimuli, and then in that case, there's a hormone kind of telling the endocrine gland to secrete another hormone. So that's kind of part of a bigger, more complex feedback loop. And then there's also neural stimuli, which is a fast response. So I mentioned that the nervous system tends to be a very fast response. Endocrine tends to be kind of a slower response to internal stimuli. But sometimes you have to have a really quick release of different hormones like epinephrine and norepinephrine. And this happens when you have your fight or flight response. So that sympathetic nervous system releasing epinephrine and norepinephrine, that's a very fast response from a neural stimulus. So when we're thinking about what those endocrine glands actually are, I kind of briefly mentioned a few of them already. I'm going to walk you through them kind of going from your head down throughout your body, so superior to inferior. And then I'm going to go over the histology of a couple of them, which are kind of important, and then we'll think about function. For lecture, you do not need to know all of the hormones that are produced in each of these glands by any means. Just kind of focus on knowing what the glands actually are and their rough locations. So starting in our brain, we have the hypothalamic pituitary axis, which has the hypothalamus, the anterior pituitary, and the posterior pituitary. So you can see those in your in this kind of inset at the top of the picture. The thalamus and the pituitary gland are towards kind of your face in your brain, so they're kind of towards the anterior part of your body, your anterior brain. We also have the pineal gland, which is more posterior. Moving down at our throat, kind of sitting on top of our trachea, we have our thyroid gland and the parathyroid glands, which are on the posterior side of the thyroid. I'll show you a picture of those in just a moment so you can better visualize that. Again, we have the parathyroid glands. Again, para means kind of next to or adjacent, so the parathyroid glands are next to or adjacent to the thyroid gland. We have the adrenal glands. The word renal means kidney, so adrenal means on top of the kidney, and you can see them in this diagram sitting on top of your kidneys. There's the pancreas and the gonads. So we'll talk about the function of the gonads and kind of the signaling molecules that are involved there when we talk about reproduction. Okay, so I mentioned the parathyroid glands are posterior to the thyroid, and you can see them here. They're pictured on the left in yellow. So we have this thyroid gland, which has a unique shape, and then the parathyroid glands are embedded kind of in between the thyroid and your trachea. So they're on the posterior side of the thyroid. And on the right, we see a histological slide. The parathyroid gland is really dense tissue. The thyroid has these follicles, and then inside the follicles, we have fluid. So the cells kind of lining the follicles and the fluid inside the follicles secrete different types of hormones. The thyroid and the parathyroid also work opposite of one another to regulate blood calcium levels, so they are involved in a negative feedback loop for blood calcium. This is a picture showing the histology of the adrenal glands. So this should in part kind of impose upon you how complex this area near your kidneys are. Your kidneys are very important for a lot of homeostatic mechanisms. And we'll talk specifically about the function of ADH, which is antidiuretic hormone, but they are involved in a lot of stuff. So here you can kind of see these different zones of the adrenal glands. So just like the kidney, they have a cortex and a medulla, which we're going to be talking about in a moment. The pancreas also kind of has unique histology. It has these islets that are kind of scattered throughout and look kind of like flowers. These have alpha cells, beta cells, and a couple of other types of cells. I wanted to highlight the alpha cells and the beta cells because the alpha cells are responsible for secreting glucagon, the beta cells are responsible for secreting insulin, and those are two very important hormones, especially in terms of kind of blood sugar levels and digestive processes. So you talked in lab a little bit about energy and nutrition and about sugar, and so we're thinking about kind of how that's regulated here. So there's a lot of other organs that are not technically part of the endocrine system but might have functions that are kind of associated with the endocrine system. You don't need to know these for the lecture exam, just kind of be aware that functionally this is very messy and very complex, and even though we kind of divide out the endocrine glands, there's other organs that are able to help with homeostasis and with endocrine function quite a bit. Okay, so in this last set of slides, I wanted to focus more specifically on the function of ADH and our kidneys to prepare you for lab next week. And I wanted to start by talking about this case study. So back in 2007 in Sacramento, there was a radio contest called Hold Your Wee for a Wee. And so basically this radio station was like, you need to drink the most amount of water and not use the bathroom in order to win a Nintendo Wii. So there was this mom, she was 28 years old, her name's Jennifer, and she participated and won the contest, but then she died from a condition called hyponatremia, also kind of referred to in relationship with water toxicity or water intoxication. So she drank around 2 gallons, which is 7.6 liters of water, and then died within a few hours. So she called her coworker, said that she was not feeling well, that she was going to go home. She was in extreme pain and she had a very intense headache. So that headache part is very important because it points to the mechanism for how she died. The radio station got in a lot of trouble, and it was also really problematic because when they were doing this contest, people were calling in being like, hey, your contestants could actually die, this is very serious, and people had died in very public ways from hyponatremia in the past. The radio station just blatantly didn't care and was like, you know, they signed an agreement, a liability form. The liability form was for more like PR situations, not necessarily for someone actually dying. So they were ultimately held negligent, and they, the family got ruled in favor, or sorry, the jury ruled in favor of the family in the situation. Okay, so thinking about hyponatremia, briefly I wanted to review diffusion. So remember, when we have kind of different solute imbalances, we have different flow of water through osmosis in response to that if the solutes themselves can't move quickly. So in the top right, if we have a balloon representing a cell in an isotonic solution, water's going back and forth, everything's cozy. If the balloon or the cell is in a hypertonic solution, so there's more solutes outside the cell, water is going to leave the cell to go dilute those solutes, and the cell is going to shrivel up. In a hypotonic solution, water is going to move into the cell to dilute the material inside the cell, and that cell is going to expand. Eventually it might burst, but in certain situations, there are structures in place that prevent it from bursting. So if you consume too much water and you don't urinate, that leads to your interstitial fluid, the fluid in your body, being too dilute. So it's hypotonic relative to your cells. So water is going to flow into the cells. It's going to flow into your body cells to balance this out. And that happens everywhere in your body, including inside of your brain. So while the rest of your body can kind of expand, even though it might be uncomfortable and lead to distension, your brain is surrounded by your skull. So there's only so far it can expand, and if it expands too far, then you increase intracranial pressure to really deadly levels. So as your brain swells and it presses against your skull, that leads to brain damage, it leads to very serious pain and other symptoms, and eventually to death. So that's what happened in this woman's case. So this points to this idea that if we are not urinating, if we're not engaging in these homeostatic mechanisms, it can lead to really serious issues with our body. So I wanted to focus on that renal urinary system for just a moment and talk about the anatomy and the function. So when we're thinking about how our blood is filtered, how we go through osmoregulation, kind of the balance of water within our body, that's accomplished using our kidneys. So blood is filtered and urine is produced in the kidneys, and we have two of those. They're towards our back, so when you were dissecting the fetal pig, you probably saw them towards the posterior portion of the pig. That's also why if you're watching like MMA and someone gets a kidney shot, it's on their back. Those kidneys are connected to ureters, which are tubes that transport the filtrate, which is now urine, to the bladder. The bladder stores that urine until it's ready to be released through the urethra. In people with penises, that urethra goes through the same pathway as kind of the seminal fluid. In people with vaginas, it's separate from the reproductive anatomy. So focusing first on the kidneys, I don't know if you had the opportunity to dissect the fetal pig kidneys or if you've looked at kidneys in the past, but when you dissect them and you kind of do a longitudinal cut and open them like a book, you see two different zones. On the outer edges, there's the renal cortex, and then inside there's the renal medulla. And so there's some kind of structural and functional differences there, and we have different chemical gradients taking place. So as blood enters the kidney, it's entering through the renal arteries. Remember, arteries move blood away from the heart to body tissue. So blood is entering through the renal arteries, it's going to get filtered, and then it's going to go back to the heart, eventually through the renal veins. So there's tons of blood coming in. That blood is not just coming in the renal arteries though. Those are getting smaller and smaller, getting into arterioles and then capillaries, and special capillaries called the glomerulus. So those are going to interface with the kidney. The kidney is made up of all these like packed together tubes, which are called nephrons. So humans have a huge amount of nephrons, anywhere from 200,000 to 2.5 million or more, about 1 million on average. And that number fluctuates depending on demand, and actually when you first get pregnant, your number of nephrons increases dramatically because you have very different physiological needs, so that changes your physiology right away. So this number is flexible. And the nephron is where all the filtration happens in the kidneys. So in the nephron, we have kind of the interface between the circulatory system and the nephron, and that's the renal corpuscle. In that, we have the glomerulus, which is that network of capillaries that comes in, and basically that blood is flowing, and anything that can leave the capillaries leaks out at this point and goes into the nephron. So really tiny stuff is able to leave that glomerulus and go into the nephron as filtrate. And it enters at a place called Bowman's capsule. So this is a protective sac that's around the glomerulus, and so it allows for filtration based on size. So basically water, glucose, salt, amino acids, tons of different ions, urea, so different nitrogen waste products, pass through into Bowman's capsule, into the nephron. Bigger structures like cells, platelets, larger proteins don't pass through. And actually, if you have proteins in your urine, that's a sign of glomerular damage. So once that material passes through, it's no longer considered blood, even though it originally came from your blood. It's called filtrate. So it's in between blood and urine. So there's a lot happening on this slide, and when I teach this in lecture, I have the luxury of kind of diagramming it out very slowly on the board. I will try to make a video of myself doing that if I can get the resources in time, and film myself doing that, or I might just find a video on YouTube for you guys to watch. Anyway, when you're looking at the kidney, remember along the outer edge, there's the cortex, inside there's the medulla, and so if we kind of take a chunk there and flatten it out and look at the nephron that's spanning that cortex and the medulla, we see that different structures of the nephron are found on different areas of the cortex and the medulla. So remember, we're starting up at the top left where we see that glomerulus and that Bowman's capsule that we just saw. And so once filtrate enters through Bowman's capsule, it's going to go what's called the proximal convoluted tubule. Here it just says proximal tubule. That's just a fancy way of saying nearest twisty tube. So proximal means close, convoluted is like twisty or confusing. Tubule is a fancy word for tube, so it's the closest twisty tube. And so at this point, our body is going to start reclaiming some of that stuff that was in our filtrate that we want back, because we want to get rid of stuff like different drug byproducts, different waste products. We want to get rid of those, but we want to take back bicarbonate and salt and different nutrients, different ions and water especially that we don't want to quite get rid of yet. So then that filtrate's going to keep traveling, and we haven't quite reclaimed everything that we want at this point. It's going to go through the descending limb of the loop of Henle. So this big loop is called the loop of Henle. Descending, we're going down, and the descending limb is actually permeable to water. So we're able to take back a lot of water, and that helps create a concentration gradient. So now the medulla is very watery, and the inside of the nephron, that filtrate, is very, very salty. So we have a ton of salt inside of the nephron relative to the outside. So we're able to kind of passively allow salt to diffuse back out. The ascending limb of the loop of Henle is not permeable to water, so water's not passing back and forth. So we're able to kind of passively take back salt and then actively pump it out into our medulla. And then we get back up to the distal convoluted tubule. Distal is like distance, so now this is just the farthest twisty tube. So at this point, we're still going to be reclaiming some of those nutrients and salts that we might have still in the filtrate. Then we get down into the collecting ducts. So we have one last chance to take back salt and one last chance to take back water. And the amount of water that we take back in the collecting duct is controlled through hormones. So I wasn't going off on that for no reason. We're bringing it back to hormones. So in the collecting ducts, we have these aquaporins. Aqua means water, porin is an opening. So these are channels or openings that allow water to pass back into our body. And the more aquaporins we have in that collecting duct, the more water we're able to take back. So we're able to regulate the number of aquaporins using a hormone called ADH. This is what it looks like. ADH stands for antidiuretic hormone. So anti means against. Diuretic is something that makes you pee. So something like caffeine or alcohol or a lot of different types of medications are diuretics. They make you increase the volume of your urine. And a hormone is a signaling molecule. So antidiuretic hormone is a signaling molecule that makes you not have to pee as much. It helps you build more aquaporins so you can reclaim more water. Your urine is then more concentrated and you're able to keep enough water. Levels of ADH rise at nighttime, so that's why, as we get more used to our bodies and to these regulation systems, we're not having to constantly get up at night. But when you're little or very elderly, your ADH cycles change, and so then you have more urine at nighttime. And there's a lot of other factors that affect ADH production as well. So we're going to talk about the feedback loop involved with ADH. So again, more ADH means more aquaporins, which means more water for your body, less water for your urine. Okay, so to summarize this, we have what are called osmoreceptor cells in our brain, in our hypothalamus. So osmo, they're sensing kind of water and salt levels, and so they're able to detect when the solute concentration in our blood serum is too high. So that means that the water concentration is too low. So they're going to send a signal to the posterior pituitary gland, which is then going to release ADH. ADH is going to travel through our body and bind to the nephron at the collecting ducts. Or technically at the distal convoluted tubule, but close to the collecting ducts. That signal is going to cause more aquaporins to be put into cell membranes. So water is going to get reabsorbed into the body. That means that the urine is going to become more concentrated. Our blood serum is going to become more dilute. So then a signal is sent to the hypothalamus that stops this process. Those osmoreceptor cells recognize that blood serum is now being dilute. They stop sending that signal to produce ADH, so it's a negative feedback loop. So again, negative feedback loops keep us in homeostasis. Okay, so I'm going to post the other lecture hopefully by tomorrow on digestion. Please make sure you complete the quiz from last week's material and look over that quiz feedback. The next quiz study guide will be posted on Friday. Make sure you're comfortable with it and email me if you have any questions. I'm going to try my best to catch up on grading your reflections this week. I know that I'm behind on that, but there's going to be another one due on Monday, just like always. And then please make sure you complete the next quiz between Monday and Wednesday of next week. I have the quizzes open still to allow you to look back at your feedback, but I can tell on Canvas when they're submitted late. So before grades are submitted, I'm going to have to go through and remove all of the late quizzes unless you got permission from me to turn them in late. So just keep that in mind when you're calculating your grade and let me know if you have any questions.